Good afternoon, class. It is 12:58. It is November 16, 2018. My name is Mike Gonzalez, and we're going to go over gram staining here. I just completed part 2 of week 3's lab, and here I just have some of the tools here that we're going to need to do a gram stain. Not going to physically do the gram stain, just going to walk you through the steps. And you just need some basic things here. You need a lighter of some sort, obviously, to light your candle. I use a tea light. And then I also use a test tube grabber to hold on to your slides. Then in this particular case, we needed 3 different slides, blank slides, and then a Sharpie pen. And then you also need a basin here for your dyes, and then an inoculation tube, loop, to spread your microbes on. So here, I have them lined up. So here you have your slides. So here's my broths. They're refrigerated. Put them in a test tube rack. They are labeled, each one. I'll show you. A lot of sediment on the bottom here. And you just put your inoculation tube inside. Of course, it's stained. It is cleaned with alcohol. You just insert inside the tube after you run your lips through the tube lips through your candle here. Just sanitize it, and then you just inoculate it. You put them on these slides that you already had labeled before. Also do a little circle on them to make your target spot. You swab into the middle of these circles. You let them air dry, and then you fix them through here. The reason why you want to do them through the fire is because it fixates the bacteria cell wall, and so that way you can stain it and see it through your microscope. When you go through these, you're gonna do your crystal violet, which is number 1. This one you're gonna do for 60 seconds, 1 to 2 drops. Then you'll wash off in tap water. You'll go to your iodine for also another 60 seconds, and that's 2 to 3 drops. And then you go into your 30-second testers. You go to the decolorizer. This is for 30 seconds. You're going to do also 2 to 3 drops. And then the last dye you're going to go into is number 4 here. And this one is also for 30 seconds, and you also rinse off after each and every one. Well, then you ask, well, what is your result? So here's my E. coli. It's right here. And within the E. coli, they have little specks here. You can see the one that you can see really well through the naked eye before you put on the iodine is our epididymis, as you see right there. And then you go into the cervix, which is really tiny. They're really actually hard to see. I don't think you can see it through the lens here. But they're really tiny. These were really wonderful to see under the lights, or through the light in your microscope. It was really interesting. So that's just a really quick synopsis of gram staining, the whole little process. If I walked through the actual process, it would take a while. But it was really neat. Something I've done in the past, but it had been a while. It was real interesting. Anyway, so everybody have a blessed day, and have a good day. Bye.